Welcome back to another session of RNA biology, and we were here in last class about alternative mRNA processing, and it can do in 4 different ways. And one is, the name itself says, alternative processing of different promoters. We will see them in detail, and utilization of alternative polyadenylation sites, and then alternative splicing, that means utilization of differential exon intron boundary. That means it can incorporate few bases from the intron into the exon, or it can include some part of the exon as intron. So different ways are possible. And then lastly comes the RNA editing, that is, you are changing the existing RNA to perform something else. So all these ones we will see them step by step, because it is quite interesting and it's quite exciting. You realize that how systematic the world has evolved, and how systematic or high intriguing the evolution has taken place at the molecular level. What is alternative processing? Alternative mRNA processing is the conversion of pre-mRNA species into more than one type of mature RNA. And if it gets converted into more than one type of mature RNA, you end up getting more protein or little bit different protein. When you say different protein, that doesn't mean that it is entirely different. Say if a protein have got 100 amino acid, you make 10 amino acid change, then technically it is a different protein. So I am not saying that in order to have a change, you need to have 10 amino acid change. Say one simple example I will tell you. All of you would have heard about sickle cell anemia. Sickle cell anemia is a condition in which the RBC, red blood cells, can form the shape of sickle. Normally we know it is a concave circle, like it look like a circle, little fat circle, which has got central portion little bit pressed, that is little bit concave on either side. So that is the typical shape of the RBC, red blood corpuscles, which is the hemoglobin bearing molecule, because of which you are able to deliver oxygen to various parts of the body. So in the sickle cell anemia, the RBC, instead of their circle shape, it can become shape of a sickle, means extremely elongate, and it can form the shape of a sickle. Sickle is basically a circular sword which you use for cutting weeds, etc. Farmers use it. That is the, you can look about how a sickle will look like. It is like semi-lunar shape or something, you can say, but it's definitely not circle. It has got huge implication. It can clog your coronary arteries, and you can end up getting a heart attack, etc. when you have more sickle cell shaped cell. However, a RBC will become sickle cell shaped only when there is a reduced oxygen tension. Like if you go to high altitude, like this was initially discovered during the First World War, when in the First World War or Second World War, I don't know exactly, but one of the World War, when pilots were flying in very high altitude, suddenly they were, they are getting a cardiac arrest. And then when they analyzed their blood, they found out that the reason is sickle cell shaped cells in their bloodstream. It is something more prevalent in Africa, and also in India. It is prevalent in people who are living in some endemic groups, especially who are like some types of tribes, etc. It is prevalent, and there is also can be done some screening for sickle cell anemia. But now, how this is possible? A single amino acid mutation, that is a place where glutamic acid should be there, that place a valine comes, and one single base amino acid change can create havoc in the structure of the hemoglobin, and that can create the death of the individual if they are not lucky. However, it comes with an interesting advantage. The malaria parasite, which grows usually in the RBC. So, but if you have a sickle cell RBC, the malaria parasite cannot grow inside that. It is somehow find it less attractive or do not able to grow. So what happens in a malaria endemic area, those individuals with the sickle cell RBC will be able to make a better living. So you can understand, a malaria endemic area, the sickle cell shape, or means don't think that any given time they will have the sickle cell shape. Only when there is low oxygen tension, when there is low oxygen, then only this gets, otherwise the hemoglobin or the shape of the RBC look the same. So but the malaria parasite is not able to grow. So there is a trade-off. You have a sickle cell phenotype, but you get advantage over malaria infection. Your malaria parasite will not be able to kill you because of the sickle cell RBC. You still have some RBC left which is resistant to malarial infection. So this is a kind of trade-off. I gave this example just for you to understand that even if one amino acid change is there, and we will see some example with RNA editing. Many times RNA editing happens at one amino acid or one base level, which may bring in another amino acid, because of which you end up getting a advantage, and some of this editing are so important that the organism will be able to live. If the editing is stopped, organism will die, embryonic development will not happen. So it is so crucial. So alternative processing means it can cause change in the amino acid sequence, eventually the amino acid sequence in the when the organism is doing the protein translation. Okay, so alternative mRNA processing is the conversion of pre-mRNA species into more than one type of mature RNA. The purpose is quite clear. You should be able to produce diverse proteins. And types of alternative RNA processing include alternative or differential splicing and alternative or differential poly A processing. Both can give rise to differential outcome. That means a tissue A and tissue B you have. Because of this differential processing, tissue A can have protein A and tissue B will have protein B. However, both have come from same gene. They are not coming from different gene. So it's a clever way of enhancing the complexity. One more thing I would like to tell you, when human genome is sequenced, it is assumed that around 23,000 genes are there. But a complex organism like human to have around 1 lakh genes are supposedly be present. So 23,000 is only 1/4 of 1 lakh. So where from that extra 75,000 genes are coming from? So it is believed this alternative way of producing different types of protein could be the only way in which you can have variety of proteins and you will be able to perform. Just like remember about the Swiss Army knife example which I gave in the previous class, you are able to perform multiple tasks with one mRNA. Means multiple proteins can be produced with one mRNA. So those who are interested can read this article, that is ending the message, polio signals then and now. So you will know more in detail how polio signals are effectively made use of in the diversification of a RNA species from a single gene. So what are alternative polyasites? So same pre-mRNA contain more than one polyasite. Polyasite we know AUU, AAA or AAU, AAA. So this is the polyasite based on which the poly A polymerase will decide which area onwards they should be start adding the poly A tail. So it is basically an identification mark also for the endonuclease. Around 18 base pair, 16 to 18 base pair downstream of the poly A site, there will be a cleavage and the RNA will be released, which will be acted upon by the poly A polymerase. And the poly A site, they may be used under different circumstances to generate mature RNA. Now, in a simplistic term, we can say a gene have got 3 poly A site. One is say 50 base downstream of the stop codon, another may be 100 base pair downstream of the stop codon, another may be 200 base pair downstream of the stop codon. So now the question is, will it use the 50 base one, will it use the 100 base one, or will it use the 200 base one? The answer is, it depends which tissue you are talking about. if the first site is masked somehow, like we saw the raw protein binding onto the RNA, like that many proteins can bind onto this polyaside and mask it. If it is masked, then that will not be recognized by the endonuclease and there will not be any cleavage. So it will skip it. So it will keep transcription, the transcription will continue and it will encounter the second polyaside. If that is not masked, then that will be recognized and it will be cleaved. What if that also masked? Then it will keep going for the third one. So remember, if the third one is recognized, you have 200 base pair untranslated region between the stop codon and the polyaside, which is actually worked. Actually, it could have had a 50 base pair UTR, but that was skipped, and it could have had a 100 base pair UTR, that is also skipped. Now you have got a 200 base pair UTR. Sometimes this extra UTR comes with a burden also. Sometimes this extra UTR comes with a blessing. Let us see what is the burden, what is the blessing. Burden is if this UTR contains some of the microRNA recognition element. Like I told you, we will discuss more in detail about microRNA, but remember, microRNA and siRNA normally find their target. They are of around 22 base in length. siRNA or microRNA, they are around 22 base in length. They can, if they find a target on some mRNA's untranslated region, then it can cause the degradation. So basically, it is a red flag for the mRNA's survival, because extra UTR is there, extra UTR brought in extra nucleotide sequence, and extra nucleotide sequence may or may not have target for a siRNA or microRNA. This is one way. Another way can be, advantage can be, this UTR, extra UTR may provide some special secondary structure which will provide protection from the degrading molecules, degrading enzymes. So this will be beneficial. So we cannot say long UTR is bad, or we cannot say long UTR is good. The simplistic way we can say is that it depends which RNA you are talking about, which gene or which tissue you are talking about. So the system evolves in such a way that with minimum, with less, you can do more. So that is the whole agenda or the whole principle lying behind alternative polyaside. In one cell, the stronger polyaside is used by default, but in another cell, a factor may prevent the stronger site from being used. Like I already told you, one site, two site, three site, four site, there can be multiple sites are possible. Now, alternative splicing simply means the generation of different mature mRNAs from a particular type of gene transcript that can occur by varying the use of 5' and 3' sites in 4 different ways. So I told you that you can include an exon into intron, or you can include an intron into exon partially to enhance the complexity, and it can also skip. Sometimes one whole exon can be skipped, or sometimes one whole intron can be skipped. We will see examples how it is done. One way is using different promoters. Promoters, we know that they are regulatory elements which tells the system here, what I mean is the transcriptional machinery like polymerase and other factors, that where onwards the gene is situated, from where onwards the transcription should start. So this is what the promoter or regulatory element. Other than that, promoter or regulatory elements means nothing. It is basically a guidance. Like people say you can see Everest or the Great Wall of China from space. So which basically indicates a human construction. If it is a Great Wall of China, it's only an indicator. Like that, the promoter elements also an indicator. It can be anything. Who knows? Maybe another larger, huge, big structure also will be seen. That is not the only big structure seen from space. There can be other huge structures that also can be seen from space. So we should understand these indicators can vary. That means, for simple example, for a given gene, maybe from the start point, transcription start point, say some 50 base upstream or maybe 100 base upstream is good enough to act as a promoter. For another gene, it may be maybe 10,000 base upstream has got a recognition sequence. It depends on who is the recognizing factor or which protein is the recognizing factor and what gene you are talking about. So we cannot generalize. When you talk about individual genes, we cannot generalize. Every gene should have 1 kb promoter or every gene should have 10 kb promoter. We cannot say. That is why when you are trying to express a transgenic line, when you want to clone the promoter of a gene, nobody knows exactly how long the promoter is. Best is more is always better. It is safer. Like you may, if any of you are following the epic of India, that is the Ramayana, there is a story where the Lord Hanuman brings the whole mountain instead of bringing the medicine for which he has been sent for. So if you have the whole mountain, one can always climb happily and search for the actual medicine which you have, instead of searching a medicine and bringing a wrong one. Like that, if you have a big clone, big chunk of the regulatory element, there is always a chance that you have included all the necessary element. Sometimes it may fall apart. You may go into say 20 kb region, 20,000 base pair. Sometimes you go for 50,000 base pair. For some genes, it will go to 1 million. 1 million base is a lot, 1 mega base. So that can be tricky. But thing is, it is rare. It is not that common. Usually, as a thumb rule, you can say every gene have got its promoter or regulatory elements within some first 10 kb or so. 10 kb upstream of the transcription start point, it has got all the necessary element. However, system make use of different promoter. Say a gene is there. We are talking about gene A. So gene A is needed in the skin of an individual. Gene A is needed in the say thymus of an individual, and gene A is needed in the brain of an individual. Now, how will you regulate these three genes in these three different tissues? So it make use of alternative use of the promoters usually takes place. We will see them more in detail. Another way is by using different polyasides, which we kind of already discussed. It can use a first polyaside, second polyaside or third polyaside. And then third, by retaining certain introns. That means it is including certain introns as part of the coding region. And then retaining or removing certain exons. So these are all the way in which it adds up to the complexity. Let us see how is it done. See, you can see in the first version, that is A, that is utilization of differential promoter. So you can see here P1, that is the promoter 1, and then P2 is promoter 2. So now you can see this arrow indicates the transcription direction of transcription, and second one also is direction of transcription. Now let us see how is it happening. Say in a given tissue, let us talk about skin. You have promoter 1 and it is transcribing here, and this what you see in the box is ash color. 1 is the exon, first exon, then it has got second, third, fourth, like that multiple exons are there. Say if this promoter is used, it make use of this entire stretch as a single intron. So for this promoter, this is first exon and this is the second exon, not this one. Second exon, this is the third exon. So when the splicing happens, you end up getting this less colored first exon, and second and third is the same. Whereas if the second promoter is utilized in another tissue, which was part of the intron of the first promoter and first gene. Now the intron of this downstream of this first exon, you have got the promoter 2, and a part of the intron is now acting as the exon, and the exon 2 and exon 3 are same as the first one. So you end up getting this strong stained box, and you have got these two exon 2 and 3 the same. So you end up getting one totally different RNA and totally different another RNA. Remember, the second exon and third exon are same, but first exon is completely different. It can have totally different functionality of the protein. Then you can see another example, use of differential polyaside. How is it done? Here only one promoter is there, exon 1, exon 2, exon 3. So what happens? They splice together. Exon 1, 2 and 3 normally happen because the polyacide is made use of, and you end up getting one product. Remember, exon 1 and exon 2 are identical, exon 3 is unique. Now, same promoter is driving the transcription, but it did not use this polyacide, it skipped it, somehow missed it, some protein masked it, and then the transcription is continuing and it encountered another exon, and then downstream of that it has got another polyacide. Now the question is, first exon, second exon, and third becomes fully part of an intron, and whereas this third one of the first gene is now become part of an intron, whereas the fourth one, identical fourth one, is acting as a third exon for this gene, as you can see here. So you end up getting two identical first exon and second exon, and the third exon is unique. In one gene, the third exon is actual third exon. In another gene, fourth exon is the third exon. Fourth exon is coming in the place of third exon because the actual third exon is included in the intron. So in the first case, alternative use of the promoter, you had a diversity in the beginning part of the RNA, and which will in turn bring a change in the beginning part, that is the N terminal of a protein. Whereas in the second case, you will have variation in the end part of the mRNA, hence you will have variation in the C terminal of part of a protein. C terminus of a protein will be different. Now the third example. What is third example? It is making use of differential utilization of, you saw here, retaining certain introns. So now you see here, promoter is mentioned here. You have exon 1, exon 2, exon 3, and it is giving rise to one product here. Now as a variation, what you end up getting, you are getting exon 1, exon 2, and it has now retained a portion of the intron, and then you have got the exon 3. So what happened? You got a longer exon 3, and this will add up portions of an intron. So you can see here in this case, this spliced here, this spliced here, and you got this product. In the second case, this spliced here, and this did not immediately splice, but it included a portion of this intron, which was completely removed in the previous case. So you end up getting a extra sequence from the intron. And then what was the other one? Retaining or removing certain exons. So that is what you are seeing in the last example. That is, you have the promoter, you have the first exon. You can see here top, first exon, second exon, third exon, and fourth exon. Everything happens smooth. Not this ash colored box, because that has got some importance. That is skipped in the second case. First exon, second exon, it did not go into the third exon because the third exon is included in the intron. So you have first intron, second intron, third exon, and third intron is now acting as a one giant intron. So you end up getting first exon, second exon, and fourth exon. Third exon is skipped. So this is the way it will bring in extra diversity. So let us quickly think that what is the benefit of having intron? From now, we know that introns are important because introns have got plenty of roles to play. One more thing I would like to tell you. If you are making a transgenic animal, say you are making a transgenic mice, so every gene have got exons and introns. So normally if you are making a gene from the cDNA, it is already spliced, like you make RNA, you reverse transcribe it, you make the cDNA. This is a protocol in the molecular biology. So the advantage of cloning a gene from the cDNA, you are devoid of introns. So it is full gene, it's ready to go. This if you use for making the transgene, then the expressivity of this transgene in the embryos is highly compromised. On the other hand, if you include the introns, so you cloned a gene with its full introns and you made a transgenic animal, expressivity of that transgene is 100-fold higher, 100-fold higher than using just a cDNA alone. So what it indicates, introns contain elements that can accelerate the expressivity of a transgene. So naturally it will influence the expressivity of that gene when it is intact also. So one benefit of genes with introns is a phenomenon called alternative splicing. If you don't have introns, you don't have variety. It is just like if you have lots of money, you can spend nicely, but all you have is 10 rupee. So you are limited. Maximum you can have is 1 tea. You cannot think of going for a tour, vacation, etc. So introns give this luxury of introducing diversity. A pre-mRNA with multiple introns can be spliced in different ways. If it has only one intron, you are limited with just one way of splicing. Otherwise you can't. If it have got 10 intron, it is a huge variety is possible. This will generate mature mRNAs with different combinations of exon. As long as you have different introns, it gives you opportunity to explore different ways. This variation in splicing can occur in different cell types or during different stages of development, because in a given cell, if a gene is expressed, it will not keep playing around. It has a routine way. It is just like you have a defined way of making chapati using atta, you will do only that way. On the other hand, if you are making chapati with besan or puri with besan, you will not follow the same procedure. It may have slightly, or if you are making paratha, you will make a different procedure. Same logic applies when you have a different cell type, then it will employ this alternative splicing approach. If you have, say, liver cell, one particular liver tissue, gene A, it will not do all possible combination of alternative splicing. It will stick with one. But if the same gene is expressed in kidney or same gene is expressed in the intestine, it may follow another approach. But the organism have got only one gene. So this logic is what you should be keeping in mind. We will continue more in detail about this in the next class. Thank you.